Hello everyone, I am back again with a new video and today's topic is animal tissues. As we have previously discussed about animal tissues, who discovered it, what is the study of animal tissues known as like histology, BICHAT and several other things like types of tissues which were epithelial tissue, nervous tissue, connective tissue, muscular tissue and their functions also, and how is epithelial tissue connect to the connective tissue and get nutrients with the help of basement membrane. Now let's start with today's topic. Today's topic is cell junctions, which is mostly found in epithelium. Now there are four types of cell junctions: tight junctions, gap junctions, interdigitation and desmosomes. Okay, now let's start with the first one, which is tight junctions. Tight junctions basically prevents leakage of materials across the tissues, and also it checks the flow of substances across the tissue. See here, this is two epithelial cells. Okay, now there is a very little intercellular space, so when intercellular matrix flows through this, large amount of protein gets accumulated here, and when it is accumulated here, then it forms a tight junction. Okay, this is transmembrane protein. Now comes second one, which is gap junction. Gap junction basically facilitates cells to communicate with each other. It increases the rate of transfer of molecules, ions between two epithelial cells. Okay, it forms tubule like structure which increases the rate of transfer of materials. See here, this is known as microtubule and this is called as connection, not connection. Okay, don't muggle up the words, it is connection. Now next junction comes is interdigitations. We all know that digits in math means numbers, but in biology it means projections. Like here, finger like projections we will see. There are finger like processes, outgrowth or projections of plasma membrane which enter into the cytoplasm of another cell, and it provides stretchability. Basically we need stretchability in urinary bladder. As you can see, finger like projections from both the cells. This forms interdigitations and it provides stretchability to the cells, for example in urinary bladder. Now comes desmosomes. Desmosomes are also known as adhering junctions or macula adherens. Adhere means to stick, stick something to some. They perform cementing to keep the neighboring cells together. See here, there are two epithelial cells basically. Okay, now both the epithelial cells cytoplasm stretches itself inside. Okay, see this, both has stretched its cytoplasm inside it. Now when cellular matrix flows through it, it contains large amount of protein. These proteins gets accumulated in this area. As there is a large area, this is formed by protein, so it is known as protein disc. Now it gets attached with the help of tonofibrils to both these epithelial cells. See, and the part of the tonofibrils which gets inside the cell is known as rivet. Okay, now these are the four types of cell junctions we have studied about. Now we will talk about the modifications of plasma membrane. There are many types of modifications, but today we will learn about microvilli. Villi is basically found in small intestine, but here it is microvilli. It is also called brush bordered epithelium. You can see in this figure. See, this is the epithelial cell and there are finger like projections here also. This is known as villi. It increases the surface area, it increases the absorption. Absorption is basically sending back materials to blood. Okay, whenever absorption is used in biology, it is sending materials into the blood. It is non-motile, it means it cannot move, and it is non-contractile, and neither its length can change. It cannot decrease or it cannot increase. Now for example, it is found in PCT of nephron, proximal convoluted tubule of nephron. See, in nephron blood filters here. So through Bowman's capsule, it is passed through the PCT, then loop of Henle, then DCT, and then in collecting duct. See, blood contains a lot of materials which are important for our body. For example, glucose, amino acids, etc., which are important and essential for our functioning, proper functioning of the body. These are also gets filtered through this Bowman's capsule and gets into the PCT. Here, this microvilli or brush border epithelium cells are present. It increases the area of absorption, so there is 100% absorption by microvilli. So important nutrients gets absorbed by the microvilli here. Okay, so one example is this. Then is gallbladder. We all know that gallbladder stores the bile juice secreted by the liver, but the bile juice which is secreted by the liver contains a large amount of water in it. So in gallbladder there is microvilli present which increases 98 to 99% of the water and makes it more concentrated. So today we learn another function of the gallbladder. First is it stores the bile juice, second is it absorbs the water from the bile juice and makes it more concentrated. Third example is walls of small intestine. Small intestine is the area in our body which has a lot of absorption rate. Okay, it absorbs the food particles which we eat and gets digested inside the small intestine. So this is all for today. Thank you.